Okay, so what type of organic molecule is a triglyceride? Is it a protein, a carbohydrate, a lipid, or a nucleic acid? Anybody know? It is a lipid. Okay, so a triglyceride is a lipid. And this is the type of lipid that we use to store as energy. So this is what is found in your fat cells. We also, as mammals, we have subcutaneous fat that is underneath the skin, and that is for insulation purposes. We also sometimes store fat underneath our muscle and in our abdominal cavity, and that's a different type of fat called visceral fat. But anytime we're storing energy or insulating the body, it's the triglycerides. That's the type of lipid. And so remember that there's 3 long chain fatty acids here. So this would be an example of a triglyceride. Okay. So we did talk about the difference between saturated fat and unsaturated. So what tends to be the type of fat that we store as animals? Saturated. And what is it saturated with? Hydrogen. So if you look at that, there's this chain, and there's like the maximum number of hydrogen ions that are attached covalently to the carbon. So notice how that forms a really straight chain. And so what that means is the molecules can stack really close together, and that means that it tends to be a solid at room temperature. So this would be an unsaturated fat, because look at here, there's a double bond, right? And so that unsaturated fat would be an oil. So sometimes you say polyunsaturated or monounsaturated. This would actually be monounsaturated because there's only one double bond. If it was polyunsaturated, there would be more than one double bond. So if we look at the difference between trans fats and regular fats, you don't need to know this, but I just wanted to point out that a trans fat has these hydrogen atoms are on opposite sides of the molecule. So that's what trans means. Trans means opposite. And cis means on the same side. So this would be a cis fat molecule. So which one do you want to try to avoid eating, consuming? Trans fats, right? So cis fats are better for you. Trans fats are linked to cardiovascular diseases like atherosclerosis, heart attacks, and those kinds of things. Okay. Any questions about trans fats? Any questions about triglycerides? Okay. So remember that one of the characteristics of lipids is that they are not soluble in water, and they tend to be non-polar. But there is a type of phospholipid that has a little bit of polarity into it, and it is called a phospholipid. So if I drew a picture of a phospholipid just 3-dimensionally, it's oftentimes represented like this. Okay, so that doesn't have anything to do with the atoms and the bonds or anything, and just this is a 3-dimensional structure. Okay, so we call this end the head. And we call this end the tail. The head end is said to be hydrophilic. So that means that it is polar. So it has a charge to it, and it is going to be attracted to water molecules. The tail end is hydrophobic. So that means that it is repelled by water, and it is non-polar. Right, so water molecules will not cling to it, they'll be repelled by hydrophobic. So if you put a bunch of phospholipid molecules into a beaker of water, something really interesting happens in that you get a bilayer, a phospholipid bilayer. And so it looks like this, where the heads are facing out like this, And the tails are attracted to one another. So it kind of looks like this. And this would form like a bubble all the way around. So I'm not going to draw them all, but that would be like a bubble. And that layer is called a phospholipid bilayer. Okay. So there would be water on the outside of the bubble. So this would be like floating around in a beaker of water. And there would be water on the inside. So you can write that, label that as water on the inside, water on the outside. Okay, so now we have a boundary, right? Now we have like a space inside and a space outside. And this is what is actually makes up the plasma membrane of the cell. So this makes up. I'll put plasma membranes of the cell because there's, in our cells, we have internal membranes and external membranes. So the interesting thing is that this phospholipid bilayer will just occur without any like living energy put into it, right? You don't have to like have a living organism putting energy into maintaining it. It just is the properties of the phospholipids. Will orient in that particular fashion. So those are lipids, but they're very interesting lipids because they have that polar head. Okay. The next group of lipids I want to talk about are steroids. And so steroids are 4 carbon ring molecules. So they have a specific structure. And remember how I said that carbon can form into rings? And so anytime you see these 4 rings kind of stuck together, you know that is a steroid. They are also insoluble in water, but they are soluble, insoluble in water, but they can move across the membrane of cells. And they can do that because the membrane of a cell is also a lipid, right? So the phospholipids, the steroids can actually move right across the surface of the cell. So when we think about steroids, we think about male and female hormones. So this would include testosterone and estrogen. A female will have both testosterone and estrogen, and a male will have both of these, but it's the relative abundance of one versus the other that gives us the secondary sex characteristics, like big muscles or hair or growth of a beard, for example. And these are very interesting because they're not soluble in water, they actually have to be transported in solution. So they're transported via the blood, but they're transported using proteins. So proteins have to transport them, okay? So they're transported in blood, so they're hormones. But we'll put protein carriers. So they're not soluble in water, but they have to get where they need to go. And hormones, by definition, are transported in your circulatory system. So like testosterone can be produced in the testes, but it can also be produced in the adrenal glands, and so can estrogen. So we get testosterone and estrogen from the ovaries and the testes, but also the adrenal glands. So if we look at these, the structure of this is what it looks like. And so you should be able to identify this. So if I had a picture of this on the quiz on Monday, and I said, what type of molecule is this? 4 carbon rings, you should be able to tell me that that is a steroid. Notice how testosterone and estrogen are super similar in their structure. And actually, some cells convert testosterone into estrogen, and some cells convert estrogen into testosterone. So they kind of move, they can be converted back and forth. Now, you'll also notice that there is a cholesterol molecule. So cholesterol is also a steroid. So this is a precursor to the sex hormones or the sex steroids. Okay, so cholesterol is needed to produce testosterone and estrogen. Cholesterol is a really interesting molecule because it can actually be produced, and it actually also, sorry, it is an interesting molecule because it can also be turned into vitamin D. So it's precursor to sex hormones and vitamin D. And vitamin D is necessary for our bones to form. So when you hear about cholesterol being really, really bad, you know, oh, we got to limit our cholesterol, realize that cholesterol is an essential nutrient that we actually need to get in our diets. We can produce some, but we need it in our diet. And it's not necessarily a bad thing to have. You have to have some cholesterol. Maybe too much cholesterol can be bad. Okay, so the interesting thing about these lipids, like the testosterone and estrogen, is that they can be absorbed right across the surface of the skin. So you probably, or you might have heard of progesterone, which is another hormone, progesterone cream. You can also get testosterone cream, and you put it on the surface of your skin, and you can absorb. So anytime you put a lipid on the surface of your skin, it can be absorbed right into your circulation. So vitamin D is a lipid. It can be absorbed. Vitamin A is a lipid. What is another? Vitamin E, right? So sometimes you get oils that have, they're skin oils, and they have vitamin A and E in them. And the reason why you can put it on the surface of your skin is that it's a lipid, and it can move right into your circulatory system. So those are steroids. Okay. We also have lipids that are called waxes. So this is also a lipid, right? And it is water repellent. Oops, repellent. Okay. So waxes are important because they actually help to lubricate the skin and maybe prevent water loss in some organisms like insects. And you can also find waxes on the surface of fruits. So if we look at some examples here, right? So when you pick a plum, you'll notice that it has a shiny kind of, or maybe actually a dull layer, right? And you can shine it up by rubbing it, right, on your, and that's kind of shining up and removing that top layer. And so that would be waxes on the fruit to help prevent water loss. We also have honeybees that can produce wax to build their hive. So to build the honeycomb where they're going to actually lay their eggs. They actually also store food, right, in the form of honey. So they store nutrients, they lay their eggs, and they develop inside of the hive. Okay, so honey wax would also be a lipid. Now we also produce wax. Okay, so example of this would be earwax, and this has a technical name which is called cerumen. And it can be very annoying because sometimes it gets stuck in your ear. But the way that it's supposed to work is it's supposed to naturally move out of the ear and come out all on its own. And it does this, it's really interesting. The cells in your ear canal actually move, and they start and they migrate to the outside. And as the cells migrate, as they mature and age, they move the cerumen or the earwax out with them. So this is actually designed to be antimicrobial. So it's kind of a yellowish color, and it has a particular smell. Right? It doesn't, not very pleasant sometimes, but it helps to prevent infections, and it helps to like keep the water out of your ear. And they recommend, however, that you don't use a Q-tip, because if you get that cerumen or that earwax too far into your ear, it can lodge against the ear drum, the tympanic membrane, and then you can't hear very well. And then you have to go to the doctor and they have to pull out the wax, or they have to flush the ear in order to get out the wax. So that's an example of a wax that we produce. Earwax. Okay. The next big group of organic molecules, so phospholipids, triglycerides, steroids, and waxes, those are all lipids. So now we're going to talk about proteins. So this is the next big group of organic molecules. Proteins are super abundant. There's many, many different types of proteins in your body. There could be like 100,000 different proteins in your body. And they all have a specific interesting shape to them and a specific function. So proteins are composed of amino acids. Okay. So sometimes you can go to, or if you're interested in building muscle, for example, you could go down to Rite Aid and you can buy amino acids, right? You can buy amino acid powder, protein powder, right? And you can take it in. And when we take in the amino acids, we can use those because these are the building blocks. Right? If we eat a steak, if we eat animal protein, then we have all the necessary amino acids to build our own protein. So if I eat animal meat, then I'm going to be able to take that animal meat, break it down into its amino acids, absorb the amino acids into my body, and then I'm going to use those amino acids to produce my own protein. Okay? So the amino acids are the building blocks. It's a little bit more tricky if you are a vegetarian. So if you're a vegetarian, you have to eat a combination of like legumes and grains. And so in order to get the complete amino acids, you need to eat like bread and peanut butter or rice and beans, right? Those kinds of combinations give you the complete set of amino acids that you would need. So usually these are drawn looking like this. So the covalent bond between the amino acid, this is a bond, it's a covalent bond, and it is called a peptide bond. Has a special name. And so sometimes what we would call a string of amino acids, we call a polypeptide. So in diagrams and in your book, this can mean the same thing as a protein, because it's made of many different amino acids hooked together to form a polypeptide. So proteins come in two major forms. The first form is what is referred to as a globular protein. And the reason why it's called globular is because it's like a glob, right? So it's just like a very compact, highly folded structure. So it has a complex 3D structure. It is also, globular proteins by definition are soluble in water. So that means that they have to have polarity. So they're attracted to water and water can pull them apart. They are also relatively unstable, which means that they can unfold under certain conditions. So in lab this week, what was the example of a globular protein that unfolds during certain conditions? What in our lab did we look at? Remember? Not the red cabbage, that was the next experiment. It was the enzyme, right? So what we found is that when you heated the enzyme to boiling, you found that it didn't work, right? And the reason why the enzyme didn't work is it unfolded. So it went having a complex three-dimensional structure that could break sucrose into glucose and fructose to not being able to work at all because it unfolded. So what is that unfolding called? What did you find out? What word did you have to look up on your phone? It does. Denature, right? So it was denaturation. So, Asian. So these proteins have a nature, and you can denature them, right? And they come out of there, they unfold, and they don't work. So this is exactly what you do when you boil an egg or when you fry an egg. So if you think about the egg whites, they're really super viscous and thick, and that's because they contain the protein albumin. So there's albumin, lots of albumin in solution, and it's still a liquid even though it's thick. And as soon as you heat it, it denatures, it comes out of solution, and it becomes a white solid. So the denation, that's what you're doing when you're cooking an egg. You can actually also cook an egg using acid. So if you put an egg in very concentrated acid, the same thing would happen. It would cause the proteins to denature, and it would go from being liquidy to a solid. Okay. So the two things that tend to cause denaturation are temperature and pH changes. Okay. So once you cook an egg, can you ever get it to go back to the way it was? No. So denaturation, specifically with temperature and pH actually, but high temperatures has things to denature. Low temperatures tend to not cause proteins to denature. In fact, you can store like enzymes in the freezer, and they actually stay better in a frozen state than if you put them in your refrigerator. So if you freeze the enzymes and then bring them back up to the temperature, they'll still work. At the high temperatures, it does not. So if we look at some of these globular proteins, so we talked about enzymes. They speed up chemical reactions. So these would be examples. Most of our hormones are actually proteins. So for example, insulin. So when we think about hormones, we oftentimes think of estrogen and testosterone, but growth hormone, insulin, thyroxine, most of our hormones are not lipids, they're actually proteins. And they're transported in solution in the blood. Insulin is important because it helps to regulate blood glucose. We also have antibodies. And so this would be an immune function. Okay? So the antibodies have helped to protect us against infection. So these are some examples of the globular proteins that we have in our body. Okay, so if we look at why they get this shape, okay, these are the amino acids. Notice how these, you can take out a water molecule and you can form a peptide bond between the amino acids. But this is a diagram that shows why they get the shape. And so they go from being unfolded to getting more and more complex. You do not need to know the secondary, tertiary, quaternary structures. You don't need to know the difference. You just need to know that they fold. But the reason they fold is that they form hydrogen bonds between the molecules. So there would be like a little dotted line here and here and here. And those hydrogen bonding between different parts of the protein actually cause it to fold. But those are weak bonds, and so they will unfold if the temperature gets too high, if you put too much energy into it. Another example is hemoglobin. So hemoglobin is a protein that transports oxygen in our body. So that would be another example of a globular protein. Oops, I'm not done here, so I'll have to just bear with me here and put it to the whiteboard. Okay, then we have structural proteins. They are less complex, so they're less folded. So they might actually make up or be in the form of a sheet. So they are kind of sheets of protein. Fibers. They are also insoluble in water. And they tend to be very stable, so they do not denature easily. Okay. So our hair and our nails are actually composed of a structural protein. Does anybody know what protein makes up our nails and our hair, and actually the surface of the skin, because it's dead cells, but they're filled with this protein? It's keratin. Okay, so keratin, that is hair, nails, outside skin. So that is a structural protein. There is also another one that makes up the mass of our bones. So we have a skeleton that is calcified with minerals. So we have minerals, calcium phosphates, in our bone, but we also have protein, and that protein is called collagen. So this is found in bones. It's also found in cartilage. And it's found in the underlying dermis of the skin. So sometimes they recommend that you put collagen on the surface of your skin, but collagen is not soluble in water, and it's not a lipid, so it doesn't actually get in your skin, okay? So collagen isn't able to absorb, so that's why they have you take vitamins that actually help in the production of collagen. Sometimes you can actually get collagen injected in your face or where you have wrinkles, right? So that helps to prevent wrinkling. Okay? So you can have collagen injections. So the interesting thing about this is that the bones are composed of protein. They also have calcium phosphate. So if we look at bones in a little bit more detail, right, it's collagen and calcium phosphate salts. Okay. So the collagen actually gives the bone its strength in terms of being able to bend a little bit and not break. The calcium phosphate salts make our skeleton hard on the outside, right? So the interesting thing is that you can do this at home. It's kind of a cool experiment, if you have kids for science experiments, is you can take a chicken bone and you can put it in vinegar. And then every day you change out the vinegar. And so what you end up doing is you end up dissolving all of the minerals out of the bone. And then you're left with a chicken bone that you can kind of bend exactly into half. And it's like rubbery, right? And so once you get all the minerals out, the collagen is what gives the bone its strength when you bend it, right? And then it bounces back. That's the collagen. And so those would be examples of structural proteins. So just remember that, you wouldn't want your structural proteins to dissolve in water. You wouldn't want them to denature, right? And they're just a little bit less complex than the globular proteins. Okay. Oops. Last biological molecule. Okay. So this is the last major group. So nucleic acids would be a separate group from the proteins and the lipids and the carbohydrates. So Our genetic material is called DNA, and we're going to look at it in great detail. Well, not great detail, but quite a bit of detail later on when we get to genetics. But this is short for deoxyribonucleic acids, or acid. So the DNA. So DNA actually has a sugar component, and that is what the deoxyribose is. Ribose is a sugar. So when we look at the DNA, it has a sugar, right? And that's the deoxyribose. It has a nitrogenous base. And these nitrogenous bases are given letters. So what is the letter of our genetic code? What are the letters of the genetic code? Like we were watching a science fiction and they were rambling them off. Anybody know? A, G, T, C. Okay, so those are the nitrogenous bases. A, G, T, C. I think it was like the Red Planet, that really awful movie with Val Kilmer. I think it was, I don't remember. But they actually said it wrong. They said it was AGTP. I got the DNA, the genetic coding correct in that movie. Okay. So AGTC. And then we have a phosphate group. Okay. So if we look at the structure of the DNA, we can identify those three different components. So the carbohydrate, oops, this is ATP. Talk about that in a second. Okay, so the carbohydrates, the sugars, right, are the 5 ring, the carbon right here. So this is the sugar. And so that makes up part of our genetic material. Part of our genetic material is composed of sugar. So the P is the phosphate group. And notice how it has a charge to it. And then the nitrogenous base, you can remember that because it has lots of nitrogen, right? So you should be able to, if I showed you this picture and I took the phosphate, sugar, and nitrogenous base off there, if I pointed to one, you should be able to identify it. So these are the units by which the DNA molecule is built. So this is the, like the building blocks, and they actually go together like this. So this is the sugar, this is the phosphate group, this is the nitrogenous base. And so just notice that between the two strands of DNA, there's these dotted lines. And what do you think those dotted lines represent? What kinds of bonds? Somebody said it. Hydrogen bonds, because hydrogen bonding is weak, right? So what is going to happen if you heat up your DNA? It'll unravel, but it will also, the two strands will come apart, right? So if you heat up the DNA, you'll break these down, and the DNA comes apart. And you're like, why would the DNA ever want to come apart? What happens when the DNA comes apart? What is this? Makes a copy of itself, right? So when your cells need to reproduce, or when your cells need to divide, it has to make a copy of all the genetic information. So that's why those weak bonds are significant. And so we'll talk about DNA replication when we get to genetics. Okay. The other type of molecule that is a nucleic acid is called ATP. ATP. Okay. So that's written here, adenosine triphosphate. So it has a base. This would be a nitrogenous base right here, is they have adenosine. The ribose would be the sugar, and then triphosphate, it has 3 phosphate groups. Now these phosphates actually produce really high energy bonds between them. And so we can show a chemical reaction that occurs inside your body to release this energy. So we call ATP is the common energy used by all cells in the body. Okay, so it's not genetic material, it's actually a common currency. So it would be like, I have proteins, I have glucose, but I have to convert them to ATP before my cells can use that energy that's stored there. Okay. So we talked about how we can break bonds and release energy. So the way that we would write this is we'd say ATP is converted into ADP, so there's two phosphates, adenosine diphosphate, plus a phosphate, plus energy. Right, so I can break down ATP. My muscles do this all the time. They remove a phosphate group, and that they turn that energy that's released into mechanical energy, and it causes the cells to contract. Okay. So ATP is energy that's used by our cells. So those are nucleic acids. So you should know that ATP and DNA are examples of nucleic acids. Okay. So we are moving on to the next chapter. Is this chapter 3? Does anybody know? Okay, chapter 3. So we're going to start talking about cells. So remember that this is the fundamental unit of life, right? So enzymes are not living, DNA is not alive, right? We have to have a cell. Although I think I mentioned that viruses are proteins and DNA, but they have to use a cell in order to reproduce, and that's why they infect cells to do that. So if we look at cells, we can look at the two different types. So we have what are called prokaryotic cells. These lack a nucleus. They have some other characteristics where they don't have little membrane-bound structures inside. But just say pro means before, kary means nut. So this is before the nucleus. So these were actually the first type of cells that we thought that we see in the fossil record. So if the Earth is 4 billion years, then these showed up about 3 billion years ago. We see prokaryotic cells. Okay. This is as opposed to eukaryotic cells. So anytime you see "u" in front of a word, this means good nucleus. So they have a nucleus. They also have internal organelles. So these would be like, we have organs, but inside your cell, you have little tiny organs, and so we call them organelles. So they would be like synonymous with, we have a liver, you know, but they have like a Golgi apparatus. So it's just inside the cell. Okay. These organisms would be only bacteria. So we believe that bacteria evolved first, and they're really tiny, so they're quite a bit smaller than the eukaryotic cells. So the eukaryotic cells are larger, and so this would include fungi, so like mushrooms, plants, and animals. Bacteria are single-celled organisms. Plants, animals, and fungi are multicellular, right? So we're composed of many cells. So eukaryotic cells came after prokaryotic cells. So they're derived from prokaryotic cells, and we'll talk about some evidence of that. Okay, so if we look at the size of different cells. So if we look at an ostrich egg and a chicken egg, right? Those have a single cell in them. They also contain nutrients. So they also have like yolk, and they have albumin, and they have membranes. But the egg is found in them. You can see a frog egg with your eye. Okay? Human eggs are almost to the point where we cannot see them with the naked eye. So remember we talked about micrometers. We tried to estimate the size of our cheek cells. Our cheek cells were about maybe, was it 40 micrometers maybe? So they would be right here, right? So that would be plant cells and animal cells. And then the organelles and the bacteria are much, much smaller. Right? We cannot see with a light microscope atoms. We cannot see most of the larger molecules. So you would like never be able to see a protein underneath a light microscope because it is too small. Okay. So it just gives you an idea of the scale. This is a bacteria, right? So bacteria cells can produce toxins. They can also invade our body, and they're invading our body in order to give nutrients so that they can grow and reproduce. So they're organisms and they're living. So this is the diagram of the cell that you need to study, and this is from your book. So on the quiz, maybe not, I don't know how far we'll get today, but on the quiz, I'll have those all, cut out. And so you would need to know that this is the mitochondria. This is the Golgi apparatus. These are lysosomes. So this is an image you want to study. Okay. So this is a eukaryotic cell because it has this nucleus, but also notice that it has all these other structures, and those can be described as organelles. So let's look at the nucleus first. So if I was to draw my cell, or a cheek cell, you might have noticed that there was this round structure in there. That contains, or that is the nucleus. And this is where the majority of your DNA resides. Vast majority. Okay, so that's where it stays. It stays in the nucleus. So it's kind of like having a library where you can't take any books out, but you can make photocopies of pages. So that's kind of like the library. It's like it's got all the information for the cell to function, and the DNA itself never leaves the nucleus. It stays there. Okay, so it's packaged in the nucleus. This outer membrane is called the plasma membrane. And then between the nuclear membrane and the plasma membrane, this is called the cytoplasm. Sometimes it's also called the cytosol. So those two words sometimes are used interchangeably. The cytosol is filled with water, and it's got all kinds of things in solution, so it's thick. So if you think about like egg whites, it's thick, right? It's got fluid, but it's also got everything dissolved in it, so it's very thick and gelatinous. And remember that there is fluid outside of the cells, and this fluid has a name, and it is called the interstitial fluid. So each and every one of your cells in your body is surrounded by interstitial fluid. So your cells are bathed in water. That's kind of hard to see. It's stitial, so it's S-T-I-T-I-A-L, interstitial. Stitial actually means tissues. So it's between the tissues, but it's also between the cells. Every one of your living cells, I'll have to say that. Sometimes you have dead cells on the surface of your skin and there's no water between them. But living cells have interstitial fluid. Okay, so if we look at the nucleus has the DNA. And the DNA, when it is unwound, is called chromatin. So this is unwound. Right. So if we look at your diagram of your nucleus, you can ignore here this nucleolus. We're not going to talk about that. That's just a special place in the nucleus. So you don't need to know the nucleolus. So this is showing the chromatin. And so notice how the DNA molecules are just kind of dispersed inside of that. and they're directing the production of proteins on the outside of the nucleus. When the chromatin condenses, when the cell gets ready to divide and make a copy of itself, it becomes chromosomes. So chromosomes are condensed and it's packaged DNA. So you're asking, well, why is it sometimes called chromatin and sometimes called chromosomes? It depends upon how condensed it is. So a chromosome doesn't look like these strands, but a chromosome looks like this, right? So that's what a chromosome looks like. So it condenses and packages the DNA into something that looks like that. So that's the nucleus. We'll talk later about the membrane around the nucleus and how there's pores. So you can see here, there's an opening here because the information in the DNA has to somehow get out to the cytoplasm to be read. And so it goes out through the pores in the nuclear membrane. But we'll talk about that more when we start talking about protein synthesis. Okay, so this is just another picture of showing the chromatin, right? And the way that it gets packaged, we'll talk about this later too, is it winds itself around proteins. These proteins are called histone proteins. And so this is a color-coded picture of chromosomes. And you'll notice that they come in pairs. And that's because you get one set of chromosomes from your father and one set of chromosomes from your mother. The next group of structure, next structure I want to talk about are ribosomes. And ribosomes are proteins. So I'll put that they are not organelles. But they're super important because they are like little manufacturing plants. They actually read the genetic information that's sent to them, and they produce proteins. So ribosomes are responsible for protein production. So if we look at this picture, the ribosome is in pink and blue. Sorry, I guess it's pink and blue. It's kind of weird. So this is the ribosome. So there's two subunits to it. You don't need to know that, but this would be the ribosome. And it's reading, this is genetic information that's come out of the nucleus. We're going to talk about what that is in a minute. And then this is actually the production of the protein. And so you notice that that protein is called a polypeptide or peptide chain. And that is the production of the protein. So if we look at where these protein factories are in your cytoplasm. So we have free ribosomes, they're called. And these float in the cytoplasm. Then we have bound ribosomes. And bound means that they're attached to a membrane. The difference depends upon where the protein that is being produced is going to be used. So if it's floating in the cytoplasm, it's going to be a protein that is used by the cell itself. So protein will be used by the cell. Right? So let's say it's a protein that's an enzyme that's going to allow the cell to digest stuff. And so it's going to be used in the cell, so it's produced by the free ribosomes. Now, if it's attached to a membrane, it is going to produce a protein that will be secreted. So secreted means that it's passed to the outside of the cell. So let's say that this is a ribosome that's going to produce insulin. And insulin is a protein hormone that is secreted by the pancreas. And so it's secreted by the cells, and then it passes out of the pancreas and into the blood. And then it goes all over your body. So that would be one that is secreted by the cell. So hormones, lots of enzymes. We have cells that secrete enzymes into your stomach and into your intestine. A lot of enzymes are secreted as well. Okay, so let's look at the organelles that are found in our body. So one that's very interesting is called the mitochondria. And the reason why it's very interesting is that it has its own DNA. And can reproduce inside of your cells. The other interesting thing about it is you inherit it from your mother only. So all the mitochondria you have in your body is inherited from your mother, not your father. And that's because the mitochondria are found in the egg, but there's no mitochondria in the head of the sperm. So they use the DNA that's in the mitochondria oftentimes to trace relationships between like populations. So like if they're trying to figure out like how two tribes are related to each other, they might look at the mitochondrial DNA. And we'll look at mitochondrial DNA because you can actually determine what type of mitochondrial DNA you have. If you do those DNA tests where you do like 23andMe, right, and you send away your spit and they analyze it, then they can trace you back to what type of mitochondria you have and where you came from. So did your ancestors come from Africa or Asia or Europe? They can find that out by looking at the mitochondria. So you inherit from mother only. The mitochondria is where ATP is produced. So this is where cellular respiration takes place. So when we think about respiration, we think of breathing. And the reason why we take in oxygen is we use it in a chemical reaction. And so the chemical reaction where we use it is in the mitochondria. So the whole reason you have to inhale and exhale all the time is to provide your mitochondria with oxygen. So when we look at cellular respiration, we can start out with glucose. We can combine glucose with oxygen. And so this is like internal combustion. This is fire, right? So we're going to produce heat and we're going to produce energy from this. So the byproduct of this chemical reaction would be carbon dioxide plus water plus ATP. Okay. So just like your car runs on gasoline, we can run on glucose. Okay. So we are producing energy. And this ATP then can be broken down into ADP and used to do all sorts of things. Like movement, that can also be used to produce proteins. It can also be used to fuel the white blood cells that travel through your body to identify foreign and cancerous cells in your body. So it's the common currency. Okay, so it's actually because the mitochondria has its own DNA, it's actually thought to have once been a separate organism. And there is a term for the idea of how it came inside. And so this is called, the term or the hypothesis is called the endosymbiotic Actually, I think it's a theory of the origin of eukaryotic cells. Okay, so if we look at this word, you're like, oh, this word, right? Endo, inside. Sim means together. Biotic means living. So what this means is that your cells have incorporated a little prokaryotic-like organism, and you brought it into the inside of your cell, and it lived there, and now it is an essential part of you and who you are. So eukaryotic cells evolved when larger cells started engulfing smaller cells. So the mitochondria is one that plants and animals have, is an organelle that plants and animals have. What is an example of a very similar type of organelle that is only found in plants? Chloroplasts, okay? So chloroplasts have chlorophyll, right? And that allows them to be capable of photosynthesis. So these are very similar to mitochondria. They have their own DNA. They're capable of reproducing, right? But they are capable of photosynthesis. So that's the exact opposite, where CO2 and energy, light energy this time, produces glucose. The plants have chloroplasts and they have mitochondria because they need to produce sugars and then they need to use the sugar. Okay. These would be green. So if you see a green object in a cell, that would be the chloroplast because it's a pigment that is able to absorb energy. See if I can go back to my, sorry. I wanted to go back to my cell.
What page is that on?
The cell, does anybody have their book? What's the page of the cell? Page 64? Yeah. Okay. So this is specifically an animal cell. So this you would be able to identify as being the mitochondria. Notice how they kind of look like little bacteria. Their DNA is circular like bacteria, right? And so some of your cells have lots of mitochondria, some have fewer, right? If you're a sprinter, if you're a long distance runner, then your muscle cells will have lots of mitochondria because you're going to have to be producing ATP on the fly in order to propel yourself long distances. Okay. Sprinters tend to have less mitochondria because they tend to have anaerobic or metabolism, so they actually produce ATP without oxygen. So those mitochondria replicate if they are needed by the body. Yes, there's many more than just one. Although some cells are so specialized that they lose their mitochondria. So red blood cells are so specialized that the only thing they're filled up with is hemoglobin. They don't even have a nucleus. They develop and they eject their nucleus. They're so specialized that they don't have any or the organelles. Okay. So if we look at these other structures in here, you can see there's other structures that are membrane bound. And so that's what we're going to look at is those membrane bound structures. So let's look at the endoplasmic reticulum. Okay. So when I look at this word, endo means inside. So it's not the outer plasmic membrane, so it's internal membranes, and it's reticulum. Reticulum means that it is a network. So it reticulates, it makes connections with. And so there's two different types of endoplasmic reticulum, and this is abbreviated as ER. So anytime you see ER like that in your book, that's the endoplasmic reticulum. So we have what is called the smooth ER. And it is called smooth because we can see it through an electron microscope, which is more powerful in magnifying small objects than a light microscope. And then I'm going to leave a little space here, and then I'm going to put the rough ER. So it looks rough. So these are organelles, and these organelles have spaces inside of them where chemical reactions can take place. So this just actually compartmentalizes the cell, so different things can occur in different places. So the smooth ER specifically produces lipids. The smooth ER, also breaks down toxins. So a good example of a toxin that we oftentimes have to break down is alcohol, and that is done by liver cells. So the alcohol goes to the liver. The liver cells have lots of smooth ER, and they bring in the alcohol, and then they produce an enzyme, and it breaks it down. So it breaks down the toxins in the smooth ER. The rough ER, on the other hand, is rough because it has ribosomes attached to it. And remember that I said some ribosomes are bound to a membrane, and so this is the rough ER. So what type of organic molecule is produced at the ribosome? From previous, just a couple minutes ago, we talked about it. Nucleic acids, lipids, sugars, proteins. Okay. So this produces proteins that will be secreted to the outside. To be secreted. So pancreas cells that are producing lots of insulin are going to have lots of rough ER. Most cells have a little of both, but depending upon their function, they'll have more if the cell has a specific function. Okay. If you think about it, our neurons, if you take it in psychology, individual nerve cells or neurons produce proteins. And what are those proteins called that neurons produce and secrete to the outside? Dopamine is an example. What are those called? So neurons secrete neurotransmitters, right? And so they would have lots of rough ER in them to do that. Okay. The next structure is called the Golgi apparatus. And Golgi is the name of a person. So don't try to figure out what that means from a term, from a root, Latin root or Greek way. And so the Golgi apparatus is another structure that produces sugars. Carbohydrates. It also packages and modifies, let's put modifies first, modifies and packages sugars. Oh, not sugars, sorry, proteins. So proteins that are going to be released go from the rough ER to the Golgi apparatus. So this is like your packaging plant. This is like the final place you go if you're a protein before you get secreted. Okay? So the sugar and the protein, when they come together, they produce what are called glycoproteins. So an example of this would be a glycoprotein. You might remember from the last lecture, we talked about glycogen. Glycogen being a polysaccharide of glucose. So this means sugar. Glyco means sugar, think glucose, right? This is a protein. So this is a sugar attached to a protein. And one of the interesting functions of these glycoproteins is that they allow cells to recognize each other. And they're attached to the outer plasma membrane. So some cell biologists like to say that each cell is sugar-coated. Right? It's got a protein and then it's got like a sugar attached to it, and that combination tells your body that this is, oh, this is one of my cells, versus, oh, this is a foreign cell and we need to attack it. So those glycoproteins are really important in recognition. So that's the Golgi apparatus. So let's look at the, go back to the diagram of your cell, okay? So notice here that we have attached to the nucleus, we have an endoplasmic reticulum. Notice that it has these red dots on it. Those are the ribosomes. So the RUBR is always directly attached to the nucleus. The smoothie R tends to be out to the side. So the smoothie R is right here. Okay, so this is my smoothie R. Notice that it is not rough. That's where my lipids would be produced. This is the Golgi apparatus. So this is sometimes called or said that it looks like a stack of pancakes. Okay. And the reason is it's taking in proteins from the RUBR, and then it's packaging them. And notice how it puts them in a membrane-bound structure right here. And then that passes out through the phospholipid, and then we secrete substances to the outside of the cell. So you want to think of the cell as a factory, and every little part has its own function and its own little set of enzymes that are doing different things. Okay, so I'm going to stop there for today, and then we'll continue looking at the cell, but remember that you need to study for the quiz. And so only the structures that we've already talked about will appear on the quiz here.